Welcome, everyone. Thank you for joining us at the Tissue-Based Research Workflows virtual event. Our final presentation for today is Morphology-Driven High-Plex Spatial Analysis of Tissue Microenvironments presented by Nicholas Confuorto. My name is Serena, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To ask a question, click on the Q&A box located on the bottom of your screen. Type your questions and hit submit. The speaker will answer your questions following the presentation. If you have trouble seeing or hearing the presentation, please click on the Q&A box to let us know that you're experiencing a problem. Please join me in welcoming Nick. For a complete biography on our speaker, visit the Biography tab on the left. Nick, over to you.
Hello, everyone. My name is Nick Confuorto. It's a pleasure to be here today. I am the Senior Director in charge of our Digital Spatial Profiling business unit at Nanostring Technologies, and I'm here to present on our GeoMx platform, which is our digital spatial profiler, and how it can be used to do morphology-driven high-plex spatial analysis of tissue microenvironments. So for those who aren't familiar with who Nanostring is, we are based in Seattle, Washington. We were founded in 2003 out of the Institute for Systems Biology, and we develop all sorts of different high-plex digital technologies that are mostly used for biomarker discovery and translational research. So our first platform was our nCounter system, which did bulk gene expression analysis. And then in 2019, we added a spatial element to our portfolio, which was our GeoMx Digital Spatial Profiler, which allowed us to now do this morphology-driven high-plex analysis from distinct regions of interest within tissue, conserving spatial information. But the nCounter system has well over 700 boxes worldwide now. We've been in a number of publications, and it's been used for translational research, as well as discovery research to look at predictive biomarkers in immuno-oncology. And so we partnered with, in 2017, we had partnered with Merck to evaluate the nCounter platform for doing bulk gene expression analysis as a predictive tool for their Keytruda, which is their checkpoint inhibitor therapy, their anti-PD-1 checkpoint inhibitor therapy. And in this publication, we compared bulk gene expression analysis of an 18-gene inflammatory signature against their gold standard PD-L1 IHC for its predictive power. So did an 18-gene, we set out to understand whether an 18-gene inflammatory signature, which basically characterized hot and cold tumors prior to receiving checkpoint therapy, could be a better predictive tool than just the PD-L1 IHC on its own. And what we found was these 18-gene signature, these gene expression signatures, are actually really good, strong predictive tools for response to this treatment. And so in this publication, highlighting the power of these gene expression signatures as predictive tools, and better than what was the gold standard at the time. Now, as great as these gene expression signatures have been and characterizing hot or inflamed and non-inflamed tumors and enriching for a population of patients that could potentially benefit from a checkpoint therapy, there was still room for improvement even on this biomarker signature. So while cold tumors or non-inflamed tumors rarely responded to treatment, not all highly inflamed tumors responded the same way. So there was some heterogeneity even within the populations of the highly inflamed tumor patient samples. And so trying to understand that underlying heterogeneity, trying to come up with better predictive biomarkers was a mission of ours. And obviously, you know, as much promise as these new therapies have for curing and advancing treatment for cancer and oncology, there's still a lot of room for improvement. And as we strive for better predictive tools and precision medicine, we're always looking for better ways to approach this and understanding the underlying mechanisms of response to treatment or evasion in some instances. And so, when thinking about tumor biology and why maybe a bulk expression signature on its own is not enough, we have to really understand, how the tumor works, right? So, and how these checkpoint therapies work. So, when you've got an inhibitor therapy that's removing the brakes from your immune system to go in and fight the tumor itself, you need to make sure that that tumor is primed. And that inflammatory signature gives you a good understanding of what the presence of those immune cells are within or around the tumor. But it doesn't necessarily tell you whether or not those immune cells are actually infiltrating directly within the tumor itself. So when we look at these two samples here and we compare them, on the left-hand side, both of these samples, for example, have highly inflamed scores. So that 18-gene inflammatory signature would be considered high for both of these. But when we look at the sample on the left, what we're actually seeing is high stromal contamination within the sample. We're seeing immune cells that are mostly present outside of the tumor regions. And so those immune cells aren't necessarily infiltrating within the tumor, and therefore may not necessarily be as powerful once that checkpoint inhibitor therapy is received, as compared to the sample on the right, where we see high infiltration of immune cells directly within the tumor itself. And so, you need spatial tools to really start to understand things like the tumor microenvironment. Tumors are extremely heterogeneous, as we see here with these two examples. And so, just doing analysis at a bulk level may not give you a complete picture or the full story of what's happening within your tumor sample that you're interested in profiling. And so, in this instance, a spatial tool is a requirement for really understanding that intratumor and intrasample heterogeneity. So not just spatial tools, but also bulk tools that give you all of that high-plex predictive power of, say, our bulk gene expression signatures, but then also being able to conserve that spatial information as well and bring the two together. So one of the challenges with spatial tools historically has been that they're low plex. So doing an IHC or even multiplexed immunofluorescence in a lot of cases requires lots and lots of sample because of the low plex nature of those technologies. So if we want to get at all of this underlying biology, or say we want to look at those 18 genes, but we want to look at them spatially, what we wind up having to do is take multiple serial sections of our precious clinical samples to be able to get all of that spatial information at that high plex level. And so when we're looking at very precious clinical samples, or in the instances of trying to do some reverse translation where we may have bank tissue samples and we're trying to go back and reinvestigate a past clinical study, we may not have enough sample available to us. And not only that, not only is it not a great use of the sample that's available to us to be able to do all of this, but it also is time consuming and resource consuming. It's obviously quite expensive to have to take this many sections and stain them all individually. But then finally, we're also not getting all the combinatorial information that could be gained by doing all of this from a single tissue section. So ideally, a perfect technology for predictive biomarker work, especially in reverse translation where we've got bank tissue samples, would be to get all of this multiplex data, but get all of it from a single tissue section where we can get all of that combinatorial information together from a single slide. And so, this has kind of been the paradigm out there up to this point where, if you wanted spatial information, it was mostly low plex and really more qualitative in its approach. Immunofluorescence, as great as it is, and as much as we've made strides in digital pathology, there's still a lot of work to be done to really truly digitize a fluorescent, which is really kind of an analog readout, and make it a digital readout. So information has historically been low plex, not very quantitative, and then counter that with the high plex quantitative information that we can get from things like our nCounter platform, or bulk sequencing, where we're losing the spatial information. So what we want to be able to do is bring the two of these technologies together and get a really good, clear understanding of what's happening within these tissue microenvironments, and really understand what the underlying mechanisms of both disease and our potential therapies are. So what we've done with the GeoMx system is we've really tried to build a bridge between these two capabilities. And so this is an example data set of a colorectal cancer that really shows you how we're connecting the spatial information from multiplexed immunofluorescence with the quantitative digital data of say, our nCounter system or an Illumina next generation sequencer. So in this case, both of the data sets that you're seeing here, both the fluorescence image and the digital data, have come off of our GeoMx platform. These are all from a single FFPE tissue section. And that colorectal cancer, what you're seeing is we stain that section with pan-cytokeratin in purple and CD45 in green. And then what we want to do is directly compare the microenvironment within the tumor, or say region 10, to the microenvironment within, say, an immune cell enriched region, that CD45 enriched region 7. And we want to be able to conserve that heterogeneity between those regions so we can see what's happening across a whole set of genes or proteins within each of those distinct regions. And so that's the data that you're seeing right on the right-hand side there. That data is actually directly comparing the expression analysis of this handful of proteins in this instance between that tumor-enriched region and that immune cell-enriched region. And we're seeing that with that digital data that you see there, actual digital counts of the amount of those targets that are present within those regions. And we're conserving that heterogeneity and doing that all at a very high plex level. So let me talk through. What I'll do now is just kind of explain how this technology works. We've designed the GeoMx system, our digital spatial profiler, to work with whatever tissue samples our customers have available to them in the lab. So it's been tested on fresh frozen. Obviously, it's been tested and validated for FFPE, as I had just shown. But not just FFPE and fresh frozen, but also all sorts of different tissue samples that you may have access to, whether they be core needle biopsies, FFP tissue blocks that you have to section. They could be pre-mounted sections, or even things like tissue microarrays. The system gives you the flexibility to be able to select your own regions for profiling and to really work with whatever samples you have available to you in the lab. And then once you have your samples, what we're doing is just using standard immunohistochemistry or immunofluorescence protocols for staining or labeling those sections, those samples. So you'll take a 5-micron tissue section, you'll have it mounted on your slide. That section will then, you'll do all of your staining, so antigen retrieval, staining, all on that single section. And what we're going to do is we're going to label that section with a cocktail of probes. And those can be either antibodies for protein or in situ probes for mRNA. So regardless of what analyte type you're interested in looking at, the system is multi-omics, so allow you to look at both protein and mRNA. And we'll label up that section with a cocktail of these probes. And that cocktail will include probes that have these little oligo tags attached to them for profiling. And these are extremely high plex, so we can go anywhere from 10 probes all the way up to 20,000 probes. So we have great plex capability built into this system for profiling, and that's what you'll get your digital expression data from. And then, as you've seen in that previous example, we also label up that section with some fluorescently labeled probes as well for doing the region of interest selection and for visualizing our tissue on the system. And so the system itself has 4 fluorescent channels built into it. So we'll label up that section with up to 4 fluorescently labeled probes for visualization. And all of that gets cocktailed together on that section, and then that section goes on to the genomic system and is ready for profiling. I will point out the sample prep portion of the protocol is automated. So our partnership with Leica allows us to do our antigen retrieval and our staining now completely automated on the Leica system, on the Bond RX, which allows for minimum hands-on time and obviously great reproducibility as well from user to user. So once that section is labeled up with all of those probes, it will go on to the geomic system. I did mention it is a multi-omic technology. So those probes are antibodies for protein detection and in situ hybridization probes for mRNA detection. Each probe has a photocleavable linker attached to it, and then a synthetic DNA barcode. So that barcode is what we're actually reading out on. So once your tissue section is labeled, we use a UV illumination to release that photocleavable linker. And then those oligo tags are released into a liquid that sits over top of the tissue. And then those oligo tags are what are actually being read out for your digital data with the platform. So as I mentioned, antibodies for protein-ish probes for mRNA, protocols are standard-ish protocols or immunofluorescence protocols for protein, so can fit within a standard pathology workflow. And then once your tissues are labeled, they go onto the genomics platform, and then the genomic system will go through. It'll scan the fluorescence of that tissue section, so you'll see your 4-color fluorescence show up on the genomics. You'll then go through and select the regions of the tissue that you're really interested in profiling, and there are many different ways to do that, which I'll take you through in a few minutes here. But once you've selected your regions on the tissue, you hit run, and then the system goes through in an automated fashion, and it starts picking from each region one at a time. So what it does is it moves the objective. This is a microscope, so it moves the objective over to the first region. It turns the UV light on. That UV light then releases those photocleavable linkers from all of the probes that happen to be present just from that one region that you selected. Those probes then are released into that liquid that sits over top of the tissue, and then a microcapillary system goes through and just starts sipping up that liquid and then dispenses it into a single well on a 96-well microtiter plate, where each well is actually mapped back to where it came from on the tissue itself. So we're indexing each of those regions back to a specific well on that plate. That plate comes off. Each of those wells in that plate is barcoded to each of those distinct regions. That plate comes off, and then it can be read out. Those oligos are read out either on our nCounter platform for low plex, high throughput use, or it can be read out on an Illumina sequencer for higher plex, more kind of quantitative, higher plex analysis. So the system is able to work with both nCounter as well as Illumina sequencing for the readout of those oligo tags. That data that comes off of the sequencer or the nCounter then gets fed back into the genomic system and the software, and it links that digital data back to the fluorescent image and back to those regions that you had selected on the system. So it allows you to really directly connect expression analysis back to those distinct spatial regions within the tissue. And what you see there is that output on the right-hand side of your fluorescent image. In this case, that's the tonsil section, where we stain that tonsil with pancytokeratin in purple, CD3 in yellow there, and then pancytokeratin in blue, CD3 in purple, and CD20 in those yellow regions. And now what we're seeing is distinct expression profiles for each of those morphological regions within that tissue section. So you can imagine the power of this system for doing biomarker discovery. We've worked with a number of pharmaceutical companies who've been using this platform for doing biomarker discovery within their own labs. And one of those early partners with us on developing and using the system was Janssen Research and Development in Springhouse, Pennsylvania. And so Josh Roosevelt is a scientist on their translational research team. And the first thing he wanted to do when he got his hands on the genomics system was to run a concordance study. He wanted to make sure the data coming off the genomics matched with what they were seeing with their IHC pathology scoring on a number of samples. In this case, we're highlighting 5 non-small cell lung cancer tumors that they had profiled with 12 regions of interest on the genomics platform, and they wanted to compare their pathology scoring of high, medium, and low within those samples to what we were able to see digitally with the genomics platform. And what we were able to find was the digital data matched up really nicely with their pathology scoring. And in some instances, you could see really improved upon what they were able to do with just the high, medium, and low scoring with the pathologist's eye. So it's a digital platform, genomics. You can see the large dynamic range that we have here. And so for these proteins across these samples, we saw really good concordance. And in some instances, you could see maybe where a pathologist's eye would have a really hard time differentiating between the high, medium, and low. With the genomic system, we're seeing all of the heterogeneity even within those categories. And so just giving you some advancement over kind of what's been done historically over, almost a century now of pathology work and really bringing it to the digital age. So we've got, it's great to be able to do this kind of on these geometric regions where we select kind of a tumor region, or in the case of, germinal centers or epithelial layers, on that tonsil section. But obviously there's a lot of heterogeneity even within those geometric regions or circles that you could select within the system. So how do we really get at some of the phenotypic profiling that we want to be able to look at within our tissue samples? In this case, we're showing you how we do this using what we call a digital micromirror device, or DMD chip, that's built into the system itself. What that DMD chip allows us to do is finely control the UV light in little 1 micron increments within the system. So those micromirrors, there's about 1 million of these micromirrors on this little semiconductor chip. Each one of those micromirrors is individually controllable with the software. And it allows us to finely control that UV light so we can create what we call these phenotypic masks in the system for doing profiling. So I'll give you an example of what I mean here. So this is a colorectal cancer that you see in the middle. That's a pancytokeratin stain in yellow for fluorescence. And then there's a DNA counter stain in purple that you're seeing for nuclei. What we did was we have some image analysis tools that are built into the software that allows us to look at that colorectal tissue, identify or set gating criteria for what our tumor cells are. In this case, we're saying tumor is anything that happens to be pancytokeratin positive. So we're going to set a binary gating criteria that says anything pan-CK positive is tumor. And that creates that white phenotypic mask that you see in the top right-hand corner there. We can then use that mask to control that digital micromirror to only shine UV light on the white regions and not the black regions of that masking that you see. So what it allows us to do is only shine UV light on the tumor cells, only releasing the oligo tags, the photocleavable linkers, from the probes that are bound to those tumor cells. So it allows us to profile our tumor region exclusively. And then since it's a binary mask, we can actually take the inverse of that mask and look at everything around the tumor, so the microenvironment around the tumor, and profile that as its own separate region of interest. So what this allows us to do is profile tumor versus the tumor microenvironment completely separately and really understand the underlying heterogeneity as well as the underlying biology within those distinct regions of interest. Now, as I mentioned, this is built directly into the system, into the software. So in our suite here of software, you'd go through, you'd see your tissue section, you'd select the region that you want to profile, you'd set your gating criteria, and then that gating criteria would create these masks that you see here. So in this instance, doing a tumor versus tumor microenvironment mask, you can see there the yellow and the blue are your two distinct regions that will receive their UV light independently. And then as we go through different regions of the tissue, once you've set that gating criteria will follow through to those different regions as well. So you don't have to do that every time. So in this case, this is also a colorectal cancer that you see here, CD3 in purple, CD45 in blue, and pancytokeratin in green. And then we selected some distinct regions across the tissue and used that masking capability to be able to actually profile those regions individually. And so if we look at the heat map here on the right-hand side, we're actually seeing that masking is clustering the data based off of those different unique masks. So we would expect the biology to be different within those phenotypic compartments, as we're seeing here with the data. But then also seeing some clustering between things like the invasive margins that we selected, the tumor centers, as well as even some normal colon that we selected there in region 10. So just showing you the power of this technology, not only to look at like unique signatures of expression across samples, but also to look at unique signatures of expression even within a single sample. And so, I've highlighted two of the different ways we can select regions within this platform. It's extremely flexible and allows you to ask some really great, unique questions of your tissue samples. So in the case of, wanting to look at geometric regions, which could just be circles or squares or polygons, you could be asking what's the heterogeneity of expression within these different, just regions, geometric regions of my tissue. In the case of being able to do segmentation, which I had showed previously, you may want these distinct morphological profiles that you want to be able to look at, like tumor and tumor microenvironment. We also have this capability, which we call cell type specific profiling, which allows you to select specific cell types based off of their fluorescence staining. So we have some studies we've done with folks like Dr. David Rim at Yale, where he's looked at phenotypic compartments of things like macrophages and lymphocytes and tumor cells to look at distinct expression profiles and signatures and looking to see whether they're predictive for response to things like immunotherapies as well. And then, we've got the capability to do things like contour profiling, where we want to look at expression gradients across a tissue sample. Or if you're really just looking like in a hypothesis-free manner, if you want to just kind of grid the tissue section, we do have the ability to just drop grids across your tissue section as well and get kind of an unbiased, just overall look of the expression across your tissue sample without necessarily looking within specific phenotypes or compartments. And I mentioned, I've shown you some protein expression data previously. We also have the mRNA capability built into this as well. One of the nice things about mRNA is your ability to scale the plex. So we've gone up to, I think I've mentioned about 20,000 genes we can look at with things like our whole transcriptome atlas assay that we released this year. We've also built some really nice curated content specifically for oncology called our Cancer Transcriptome Atlas. That's an 1,800 gene panel that's been curated to look at things like the tumor biology, immune responses, microenvironments, high, low, and medium expressors. All of this is based off of tons and tons of publications that have been out there publishing on bulk expression as well as single cell expression of tumors. And so it's really good high curated content, specifically if you're looking at cancer biology. But if you want to just get an overarching look at, the entire transcriptome, all, protein encoding genes, we've built two whole transcriptome atlas assays, one for human and one for mouse. And like I said, those are the entire protein coding transcriptome and really great unbiased look at the underlying biology of your tissue samples, and all done on a single FFPE tissue section. So as I mentioned, both of these have been tested and validated to work both in FFPE as well as fresh frozen, and all on a single 5-micron tissue section. And you can imagine the power of things like this, once again, for looking at things like response to treatment or prediction of response to treatment. This is from one of those such studies where we were taking our regions of interest, creating these tumor and tumor microenvironment masks, and then looking at both protein and mRNA expression within these distinct compartments. And in this instance, looking to see how the biology is changing within that pan-cytokeratin enriched region, or in that instance would be our tumor region. So both on treatment and off treatment, doing something like this volcano plot that you see here, that's showing you the significance and differential expression between samples that were profiled on treatment and samples that were profiled that were off treatment, or that did not receive treatment, and looking to see how that tumor region differs between these two sample sets. I've mentioned we have our partnership with Leica around automating sample prep. I've also mentioned our partnership with Illumina for being able to read out those oligo tags on an Illumina sequencer that will work with their all of the Illumina platforms. We also have our partnership with ACD. So if you're not familiar with ACD, they have their RNAscope technology, which is a fish-based technology for doing immunofluorescence of RNA probes. And so the RNAscope will work on these same sections that we can do our mRNA profiling with. So if you wanted to fluorescently visualizing your tissue, we do have the ability to work with RNAscope as well. We have a partnership with Abcam, which gives us access to over 6,000 of their antibodies in their catalog. Abcam has one of the largest antibody catalogs on the planet. And so having access to that catalog for doing things like oligo tagging, as well as custom labeling of antibodies in case there's a specific antibody that you'd like to see that maybe we don't include in one of our panels, you can work with Abcam, and they will help you custom label an antibody from their catalog to include in our assay. And then academic collaborations and partnerships as well. We created our Genomics Translational Leadership Network last year, which allowed us to do some collaborations around specific things like breast cancer or COVID-19 research. We also have a partnership with the Human Cell Atlas, where we help support some of their research, so some of the work that's being done for atlasing at the single cell and spatial level through the HCA. We have a partnership with the Mayo Clinic and OHSU to develop diagnostics for the genomics system as well. So while the platform today is an RUO platform, obviously you can imagine with the digital and multiplex capability of this system, and as we push further and further towards precision medicine, that a platform like this could be applied in a more regulated setting. And so we're currently exploring what the development of that would look like with this partnership with the Mayo Clinic and OHSU. And then I mentioned we have our breast cancer consortium. So this is a consortium of investigators that have gotten together to come up with best practices and different ways to visualize and look at breast cancer tissue on the GeoMx platform. And so that consortium has now come together and they are publishing those best practices. So we're really excited to see some of the results coming out of that consortium that we've set up around breast cancer for the GeoMx system. But in regards to the Leica relationship, I mean, this automation of our protocol, and as I mentioned, it's standard immunohistochemistry protocols, but you can imagine, that takes some time and some expertise to be able to know how to do your antigen retrieval, and we've got protocols that we can give to folks. So, we walk you through those steps, but it can take some time. I mean, you're talking about 3+ hours of hands-on time to manually, prepare a sample and stain it. Anyone who's done IHC work in the past can understand, that can take some time. And so what's nice about the relationship with Leica is this ability to automate that on their Bond RX system. We go from about 3 hours of hands-on time to about 30 minutes of hands-on time with the Bond RX. And the capabilities of that platform to do, things like up to 30 slides at a single time really allows you to scale your work with the GeoMx system as well. So, you do your prep the day before, you'll do your staining overnight on the Bond, you'll show up the next morning, take the samples off of the Bond, put them on the GeoMx system, scan those slides, select your regions of interest, tell the system to run, it'll go and collect. By the end of the day, you'll have all of your collections done. That plate will then come off of the system. You'll do an overnight hybridization or your library prep to go into your sequencer. That data will come off of the sequencer. It'll go through an NGS pipeline that we've set up. There's either that NGS pipeline is either loaded directly onto the Illumina sequencer, or it can be, or you can run it through your own computer system that you've got in your lab. We've got the NGS pipeline that can be loaded directly onto it. Those data, that FASTQ file gets converted into a DCC file, and then that DCC file gets loaded back into the software for your data analysis. So, all said and done, it's about a 5-day protocol with very minimum hands-on time, mostly automated through there, but gives you really great multiplex quantitative data, and obviously can be done in a serial manner so that you can constantly have data coming out of this platform. So if you really want to scale up your operations, having it automated with Leica and then being able to kind of serialize the work that you're doing can really scale the amount of data that can be generated on this system. So with that said, just to summarize everything, the GeoMx platform is extremely flexible. It's the highest plex, most sensitive system out there on the market today for doing this type of high-plex spatial analysis. It gives you the ability to detect all the targets you're interested in. So, like I said, anything from 10 up to 20,000 plus genes or proteins at a time. It is multi-omics, so we can do both protein and mRNA detection. You can explore any region that you're interested in, so it allows you to ask very specific questions, or in the case of doing things like gridding, you can just look to see where the data drives you. So it's totally up to you. It gives you that flexibility for selecting different regions of your tissue. And then it also allows you to work with any samples that are available to you as well. So whether they be FFPE or fresh frozen, or as I mentioned, core needle biopsies or tissue microarrays or whole mount sections. And then, Nanostring's got this reputation of being a good high quality, of building good high quality platforms. That's our legacy with our Encounter system, and it continues on with our GeoMx platform, and any future platforms that we develop is our quality system. And so you can trust the data that comes off of this, right? It's proven, it's optimized, and it's automated. So you get great reproducibility between users and multi-sites and multi-systems. And so that allows you to once again think about scaling that data and using it in your large translational studies. So with that said, that concludes my presentation, and looking forward to any questions that anyone might have. Thank you.
Well, thank you so much, Nick, for that informative presentation. Just a quick reminder to our audience on how to submit questions. Simply type them into the Q&A box and click on the send button. We will answer as many questions as we have time for. And let's see, the first question is, is this technology currently available for purchase or through a CRO?
That's right, yeah, thank you. So you can purchase our GeoMx system. It is available for purchase. It's on the market today. We launched it back in 2019. And so if you're looking to adopt technology, certainly you can reach out to your Nanostring representative, and we can get you some pricing. Or if you're looking to just test it out, we've got some abilities to go out there and test the technology as well. But we also have service providers. So there's a network of service providers globally that offer GeoMx as a service. They are both academic and industry service providers, so a number of CROs as well as core facilities at academic centers. So yeah, please feel free to reach out to Nanostring if you're interested in testing it, and we'd be happy to connect you with the right path.
Great. And it looks like our second question is, how many genes can transcriptome?
Yeah, so the whole transcriptome assay can profile up to 22,000 genes at a single time. It does have the ability to add customization as well. But yes, both the human and mouse whole transcriptome assays are up to about 22,000 genes. That's the entire protein encoding transcriptome. and both can include customization.
Great. And it looks like this is our last seated question, unless we have some more that are coming in. The last one, how does this technology compare to other array-based or imaging-based platforms on the market?
Yeah, thank you. So there are a number of spatial genomics platforms out there today, and it's a very hot market. Obviously, we were one of the first into it with our genomics system. And so, we're pretty unique in our approach for doing profiling spatially, and that's because we have that digital micromirror capability that allows us to select regions of interest for profiling, but then also to be able to contour those regions specifically to the biology that you're interested in. So those examples that I gave looking at tumor versus stroma, the genomic system, because it's not kind of randomly gridded on there, it's kind of in its own smart way allows you to select that biology and profile it independently, as opposed to like an array-based technology where you're kind of limited to whatever falls within that gridded area. And then for the imaging platforms, we are developing our own imaging system, multiplex imaging system as well, which we've recently announced. But we do see these as very complementary, so imaging and profiling to be very complementary. On the imaging side, you're going to be limited in Plex, and that's going to always be the case because it's always going to be fluorescently based. So plexing, to go to higher and higher Plex is going to take more and more time, and eventually you kind of hit a saturating limit there. Where with profiling, the way we do it with our oligo tags, we can go up to whole transcriptome and beyond. I mean, theoretically, there's no limit to the number of Plex that we can do. So, between profiling and imaging, profilers usually at the whole transcriptome level or beyond, imagers usually more targeted, even if it's a high-plex multiplex imaging assay, it's still fairly targeted as compared to what you can do with like a platform like genomics. And then the quantitative, aspect of it, higher throughput, more quantitative, those are usually the big differentiators between a profiler and an imager.
Oh, great. Fantastic. Do you have any closing comments for today, Nick?
No, I mean, I think, this is an area that's continuing to evolve, the spatial biology market and world. And, we're here to help guide that evolution as best as we can with our tools and technologies. So if you have any questions, if you're interested in trying out the platform, if you have a great idea for a project, we do a number of different grants throughout the year. So there should be no limit, right? I mean, if you're interested in running a study, please reach out to us and we will find a way to work with you. Right now, it's our goal to really kind of democratize, spatial biology. And so we will find a way, whether that's through, the acquisition of the technology itself or through a service provider or even through like our own internal labs, we would love to work with you. So please reach out and looking forward to more partnerships and opportunities to work with the Leica team as well.
Well, fantastic. Again, a big thank you to you, Nick, and thank you to our audience for joining us today and for their interesting questions. We will address any questions we did not have time for today and those submitted during our on-demand period via the contact information you provided at the time of registration. This session webcast will also be on demand after the event. That's all for this session, and thank you for joining us. We hope you have enjoyed this presentation. Bye for now.